Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll be discussing another important topic, that is DABT topic, that is treatment of organophosphate poisoning. So let's directly dive into the question. This question is taken from ABT recertification May 12, 2000. So the question is, the treatment of poisoning by organophosphates can be very difficult. So in this video we'll be discussing about organophosphate poisoning and its treatment. So one reason for the difficulty in treating this organophosphate poisoning is reactivating the enzyme acetylcholinesterase, because the organophosphates, they will attach to the enzyme through. So we need to identify what type of bond this particular organophosphate pesticides will form with the acetylcholinesterase enzyme in this question. So we have option A, that is van der Waals bond. We have option B, that is ionic bond. Option C, we have hydrogen bonds. Option D, we have covalent bond. Option E, we have reinforced ionic bonds. The right answer will be in the green text, that is option D, covalent bond. So, um, so let's quickly revise our memory. First, we have to understand the type of bonding that we see across chemicals. So there are two types of bonding are there, that is strong bonds and weak bonds. If you look into the strong bonds, ionic bond, covalent bond, coordinate bond, metallic bond, all this will are strong bonds in between the chemicals. And coming to weak bond, we have hydrogen bond and van der Waals interaction. So you can pause the video here and see the electrostatic, the unpaired electrons, paired electrons, how one atom is forming a bond with another atom. So this is something related to chemistry. You try to quickly revise your memory with respect to chemistry. Since we have options about all these bonds like van der Waals ionic hydrogen bond, so our answer is covalent bond. So we'll focus the understanding more towards covalent bond. Why it is a strong bond? See, basic principle is that all these things, ionic, covalent, coordinate, metallic bond, all are strong bonds only. But as far as I remember, covalent bonds, no, they are irreversible type of bond. Once the bond is formed, uh, since it is irreversible, the bond breaking is very difficult. Hence, this type of treatment of poisoning, in which such type of covalent bonds are formed, it is much more difficult, much more challenging. So, and since we are discussing about organophosphate poisoning, we have to also discuss about acetylcholine. Acetylcholine neurotransmitter, try to remember, acetylcholine is a neurotransmitter which is formed in the presynaptic neuron. This is your presynaptic neuron and this is your postsynaptic neuron and this is your synaptic cleft. Okay. Whenever by the action of this choline acetyl enzyme will be there, where acetic acid and choline, that together they form acetylcholine, and next this acetylcholine will stored into this vesicle. So whenever there is action potential is generated, stimulus is there to the presynaptic neuron, then this vesicle will come and fuse to with this membrane, and it will release the acetylcholine into this your synaptic cleft. This is your neuromuscular junction, synaptic cleft. So here you have another enzyme called acetylcholinesterase enzyme. What this acetylcholinesterase enzyme will do is, it will break this acetylcholine into again acetic acid and choline. Okay. So what this organophosphate pesticides are doing is that they are blocking this acetylcholinesterase enzyme. As a result, no more breakage of this acetylcholine into acetic acid and choline taking place. As a result, in the synaptic cleft, in this neuromuscular junction, acetylcholine levels will be increased. As a result, on the postsynaptic neuron, you have receptors. So overstimulation of these receptors takes place, and that is the reason you will have excessive cholinergic actions you will be having. Like, you have to remember that mnemonic sludge, salivation, lacrimation, diarrhea, urination. So that will be there. So throughout this concept, you have to always remember about this acetylcholine, the mechanism. Okay. So let's go into the whatever I have explained, no, in the flow diagram I have kept. This is the mechanism of action of organophosphates. It is the pathway is not only applicable to organophosphates, also those who are acting on acetylcholinesterase enzyme inhibitors. So same concept will be there. Instead of organophosphates, remember acetylcholinesterase inhibitors. So what will happen? Mechanism of action is inhibition of this particular enzyme, acetylcholinesterase. Okay, this is your presynaptic neuron and this is your postsynaptic neuron. In this presynaptic neuron, what is happening? This is your this is your choline, and this is your acetic acid. So this acetic acid and choline, they combine by the action of the enzyme choline acetyl transferase. Okay, then you will form, then acetylcholine will be formed. Okay. This acetylcholine, by the method of this high H plus transporter in the vesicle, this acetylcholine gets stored. Once this stimulation of this particular presynaptic neuron takes place, action potential, this vesicle will come and fuse with this presynaptic membrane, and it will release in the synaptic cleft, this neuromuscular junction, acetylcholine will be released. Okay, so once this acetylcholine is released, it will, this acetylcholine neurotransmitter will act on this postsynaptic neuron. In this, you have acetylcholine receptors are there. That time you will have cholinergic effects will be there. So once the action is done, this whatever in the synaptic cleft, whatever acetylcholine is there, no, that has to be degraded. So that action is taken by this particular enzyme, acetylcholinesterase enzyme. Okay, this acetylcholinesterase enzyme will break this acetylcholine back into acetic acid and choline. Okay, so what this organophosphates or acetylcholinesterase inhibitors are they doing is they are inhibiting this particular enzyme. As a result, accumulation of ACH accumulation in the synaptic cleft will increase. So excessive stimulation of muscarinic receptors, since you know that cholinergic, no, we have nicotinic receptors and we have muscarinic receptors. Okay, if the action is on muscarinic receptors, you will have salivation, lacrimation, that is sludge, demonic you can remember, and stimulation of nicotinic receptors will lead to fasciculation and conversion. So that was about throughout this concept you have to remember. So organophosphates, they will inhibit this acetylcholinesterase enzyme from breaking down acetylcholine, thereby increasing both the level and duration of action. Not only the concentrations in the synaptic cleft, also how long, how much more time it is there in the synaptic cleft. That is also important. So duration of action of the neurotransmitter acetylcholine. Okay, pesticide accumulation. This is your red color is your pesticide. Okay, accumulation in the synaptic cleft, acetylcholinesterase, this particular enzyme is inhibited, okay, by this pesticide, constant activation of this particular acetylcholine receptors. Okay, so let's I have taken these materials, this concept from the that is PK Gupta, that particular textbook. So those who people are interested to read about Indian authors with respect to toxicology books, you can read. So let us understand. This is your acetylcholinesterase enzyme. Okay, on the enzyme you have two different binding sites will be there. Now coming to the question, the covalent bond, why we are discussing that, we have to see. So if you look into the enzyme, no, we have anionic site and we have esteric site, or serine you can say, serine binding site you will say. Okay, anionic site is negatively charged. Okay, and this is your acetylcholine molecule. Okay, on this acetylcholine molecule you have nitrogen with a positive charge will be there, electropositive charge will be there. Okay, so here this is COH, no, acetic acid is there, no, this hydrolysis bond breaking will be there. So let us understand. So acetylcholine, it is broken by cholinesterase enzyme. Okay, so this acetylcholine here breaking is taking place. Okay, so because of which acetic acid will be separated, choline will be separated. The positively charged nitrogen, here it is positively electrostatic positive charge is there, no, is attracted to the anionic site. This particular site it gets attracted on the acetylcholinesterase enzyme. This is your enzyme, and hydrolysis takes place. This OH, COOH bond was there, no, in acetic acid you have COOH. This bond will get break. Okay, as a result hydrolysis is catalyzed at the esteric site to form choline as well as acetic acid. So acetic acid and choline is formed. So this is the important thing, your concept you have to remember. So let's understand in detail something with respect to cholinesterase inhibitor. So this cholinesterase inhibitor, example we have nerve agent. So pause here, go back and read about World War One as well as World War Two. In during that time, there were a lot of war agents were there, warfare agents, that is called sarin are there, taboon is there. So certain examples are there. So you can read So most of the what is the mechanism of action of those particular warfare agents you read. So that is very interesting topic. One or two questions. Still today also certain homicidal or suicidal threats are there by using this type of warfare agents. So coming back to our topic, cholinesterase inhibitor nerve agent attaches to the serine site. So this is the site. Okay. If you see, this is the nerve agent. This is a structure is there. Nerve agent is an example for organophosphate. That's why you have phosphorus and oxygen bond. This is your organophosphate. This is a structure. Okay, complete structure. This one. Okay, attaches to the. This will go and bind to this serine site. Why? Phosphorus is having electropositive charge and serine is having electronegative charge. This prevents acetylcholine. Now, in generally, what in general conditions, what acetylcholine should go and bind to this particular thing. Since organophosphate, this nerve agent is already there in the synaptic cleft, this will go and bind to the serine site in the enzyme. So acetylcholine have no longer any binding site will not be there. This prevents acetylcholine from interacting with the cholinesterase enzyme being broken down. So as a result, what happens? Acetylcholine will not break and acetylcholine levels in the synaptic cleft will increase. So that is a concept. So partially electropositive phosphorus is attracted to partially electronegative serine. Okay, positive is attracted to the negative. Positive indicates the phosphorus is partially electropositive. Negative indicates that oxygen is partially electronegative. So throughout the concept, you have to remember this one: synaptic left, presynaptic, postsynaptic. Okay. So now how the cholinesterase inhibitors are working, we'll see. Okay. Transition state shows. So this is the transition state. How the bond breaking is taking, how the hydrolysis is taking. In these two things we will be seeing. Transition state shows which bonds break and which one forms. If you look into this one, delta positive, no, indicate the phosphorus is partially positive and this is partially negative, serine site. Okay, so what is happening here? The bond breaking is taking place, and if this RL group is no, if the structure is similar to that of the anionic site, there is a possibility that this will go and bind. So possible attraction if RL is approximately sized and charged. Okay, then there is a possibility this will go and bind at the anionic site. Okay, so here the bond breaking is taking place. This is the hydrolysis is taking place. Okay. So cholinesterase inhibitors, they will attach to this acetylcholinesterase enzyme. Okay. This is your inhibitor preventing the attachment of acetylcholine. Here the bond breaking is taking place. So R L O, no, this got separated. Okay. This has been removed. Now only what is happening? P double bond O, R3, R2, only O. Here hydrolysis OH is gone. See. Okay. Now this is attached to the esteratic site. Okay. That is how the cholinesterase enzymes work. Now we'll see some concept with respect to aging of this particular enzyme. So one in our question also one concept is with respect to the aging. Okay, so cholinesterase inhibitors can lead to aging. This is important. So whatever the enzyme, no, cholinesterase enzyme, it can undergo aging. Irreversible type of damage can form to that particular enzyme. As a result of which the enzyme can no longer be able to show any cholinergic stimulation. Let's understand in detail. Phosphorylated cholinesterases, okay, this one, once it is phosphorylated, no, undergo dealkylation. Dealkylation means this particular bond ROH, this will get separated. Okay, of the organophosphorus moiety. This is your organophosphate moiety leading to aged enzyme. So here the aging takes place. Once this particular alkylation group is going away, na, one hydrolysis happens. Conversion of this inhibited enzyme into a non-reactable form. Now this enzyme, once this bond breaks and this gets going attached to the serine site, no, whatever this enzyme, no, it becomes non-reactable form. No matter how much acetylcholine there in the synaptic cleft, it can no longer. So irreversible type of bonding, covalent bonding is taking place. Aging occurs rapidly when the inhibitor is, example, they have given soman, that is a powerful nerve agent. So let's pause here once again. So, and you read about this soman and this one which was used as a nerve agent, a chemical warfare agent. The odor was similar like of a camphor or rotting fruit. Okay, and it's so poisonous, so that even in minute concentrations up to 1 to 10 ml through skin exposure, it's not systemic exposure. This that is the reason when the pesticides, no, in the agriculture, farmers when they apply these pesticides, no, if they don't wear proper PPE protective, or if they don't wear mask during the application of this pesticides, there is a possibility that through skin the absorption of these pesticides, organophosphate pesticides through skin, their absorption is faster. So here generally we come across that oral exposure, but here this particular soma, no, within 1 to 10 ml exposure through skin, it is causing fatality. Okay, that is the reason one what they will do is, whenever in an emergency department, if a person who is exposed to this type of pesticide poisoning is there, first they will declothe him, they will wash him, they will remove the clothes, so that further absorption, you can stop the further absorption of this poison through the skin. Okay, so let's come back to our topic, cholinesterase inhibitors. Okay, so aging occurs rapidly when the inhibitor is SOMAN. So we discussed about SOMAN. So cholinesterase enzyme, we are talking about this enzyme, is blocked, but it can hydrolyze to three states are there, three type of things. One is slow reaction, in which hydrolysis takes place, OH will takes place. And once this, again this OH bond form means it can, so this is the reversible type of damage. Once the that particular type of organophosphate removed from the from your term, from your synaptic left, it can go back to normal state. So still it is active state only will be there, cholinesterase enzyme. Second one is regenerate with an oxime. So we will discuss about oxime, that is 2-PAM, palidoxamine, which is used as an antidote along with atropine for cholinergic type of poisoning. So that is also a fast type of reaction. Third one is aging. Aging is cannot regenerate. So that type of bond now which will be formed, it will be much more damaging. Okay, irreversible type of damage takes place. So the aged bond after addition of water, here hydrolysis takes place. First here there was still possibility. So RLOH gone away, R2OH now gone away. Now the bond will form between R3 and O. Okay, the aged bond after addition of water to this PR3 bond. Prior to aging, R2 was pulling this negative charge, this was pulling. R2 was pulling the electron away from the phosphorus. Upon it being removed during the aging process, now there is no R2H, no RLOH. Okay, these electrons are shared with O serine. This is your this one, strengthening its bond so that it can no longer be hydrolyzed. Okay, so that is about the aging of the enzyme. Okay, very important with respect to the question what we have seen. Then now in this slide we will discuss about the antidote. Here we are discussing about 2-PAM, that is 2-pralidoxamine. We seen one picture with respect to oxide, second type of damage, which is a fast type of action. No, so in the organophosphate poisoning, an organophosphate will bind. This is your organophosphate, the 2-PAM. Sorry, organophosphate will. This is your organophosphate. It will bind to the serine site on the acetylcholinesterase. Now what you are doing in order to mitigate that risk, you are adding antidote. Your antidote is your 2-PAM. So what how this 2-PAM will act, we'll see. Will it will bind just to one side of the acetylcholine, because only what site is left, no, only anionic site is left. Okay, of the acetylcholinesterase enzyme, blocking its activity. So 2-PAM is a 2-PAM and phosphorylated enzyme. So pause the video here, read about 2-PAM, which is used as an antidote along with atropine. Atropine is an anticholinergic. Cholinergic poisoning के लिए we are using atropine along with palidoxamine. So palidoxamine is used for nerve agent type of poisoning you are using. Okay, so it is 2-pyridyl aldoxime methyl chloride. It is an important antidote. It is used along with atropine whenever there is organophosphate poisoning, whether it is pesticides or nerve agent. How it is working means it is acting by reactivating the acetylcholinesterase. Whatever the damage was done, no, it is reactivating. It is again bringing back acetylcholinesterase enzyme function. Okay, reactivating the acetylcholinesterase enzyme, inhibiting by the toxins. Specifically, it will cause relieving of the muscle weakness and respiratory paralysis. So no longer paralysis will be decreased. So it is administered via either intravenous parental route, intravenous or intramuscular route. Okay, that was about 2-PAM. So let's see the mechanism once again. Pralidoxamine is able to attach to the other half. This is anionic site. This is your 2-PAM. This is your organophosphate. Unblocked anionic site of the acetylcholinesterase enzyme. This is your enzyme. Okay, 2-PAM will bind to organophosphate. Now this particular now this particular CHN, this particular thing is NOH. This will go and bind to phosphorus. That is positive charge on the organophosphate. So this complex will form now. And once this complex is formed, this hydrolysis takes place. Now enzyme will go back to its normal state. See, 2-PAM will bind to organophosphate. Okay, here this organophosphate is there. Okay, here it is forming the bond. Okay, the organophosphate changes the conformation. Now it will change the alignment of this molecule. losing its binding to acetylcholine. So it will detach from the acetylcholine to acetylcholinesterase enzyme, losing its binding to the acetylcholinesterase enzyme. Partially electropositive nitrogen, whatever this nitrogen was there, no, on 2', is attracted to electronegative site on the cholinesterase. Okay, this you have to remember. So the conjoint, whatever this complex I told you, know, this formed, no, this conjoint poison antidote, then it will unbind from this particular site. Okay, serine site will become now available for the for the acetylcholine to come and act, unbind from the site, and thus regenerate this enzyme. So now this there is no irreversible damage, okay, which is now able to function again. Regeneration of this cholinesterase enzyme takes place when 2', and this action can only happen before the formation of this covalent bond. Before the aging happens, then only you can use 2'. Okay, so poisoning happens means you have to give immediate antidote, atropine and 2'. Once the aging has happened, once the damage is done, once the what is covalent bond formation is formed, then 2', will be of no use. It cannot act. So very quickly, if you want, you can read. I have listed out the organophosphates. Okay, their examples, various examples are there. Various classes they are used, like herbicides, fungicides, insecticides, as rodenticides, warfare agents. Okay, the same thing, organophosphates. Few questions will come in the exam with respect to chloropyrifos, diazenon, malathion, and parathion. They keep asking in the ABT. If you look into the question papers, no, previous question papers, these are you will come across this in malathion, parathion type of toxicity. Okay, so pause the video. I have listed out how the mechanism is there. Okay, then something with respect to organophosphate induced delayed neuropathy, we will see in this one slide. See, delayed neurotoxicity is observed after exposure to some organophosphate insecticides act by covalent modification on the enzyme. Enzyme is here acetylcholinesterase enzyme that will refer to as neuropathy target esterase. Here NTE is, NTE will be formed, which is a neuronal protein with serine esterase activity. Okay, so neuropathy target esterase encoded by this particular gene, that is PNPLA6 gene, is an essential endoplasmic reticulum anchored enzyme involved in phospholipid metabolism and neuronal development. So NTE is primarily known as target for this organophosphate induced delayed neuropathy. So once a person is exposed to this organophosphate, if there is a neuropathy, the mechanism is through the NTE formation. This neuronal protein will be formed. Okay, this NT will act as an enzyme for the this thing. Okay, serine esterase activity, it will be shown. Inhibition of this NT causes neurodegeneration. Okay, neurons will get degenerated, while genetic mutations lead to inherited motor neuron disorders. Okay, so this is the concept. One or two questions on OPI and OPI-DN also they will be asking. So I have taken from this picture from Clayson 9th edition, page number 1066. You go and see. This is the phosphorylated enzyme. This was the enzyme here. If you see serine and anionic site, this is your organophosphate. O is binding to the serine. So this is a phosphorylated acetylcholinesterase enzyme. Once it is coming in contact with 2', this N positive is there, no, it will form bond, and now it will backward. This enzyme will get separated. This is your organophosphate, and this is your 2'. Complex is formed. Now your enzyme is free to act. Now this free enzyme will go and break the acetylcholine into choline and acetic acid, like that it will be happening. Okay, so reactivation of this phosphorylated acetylcholinesterase by 2'. 2' is only effective before phosphorylation enzyme has undergone an aging reaction. Once the aging reaction is done, 2' is of no use. So that is very important here. So hope from this video you were able to understand about the organophosphate, the mechanism. We discussed about different types of bonds, why covalent bonding is important, why aging of the enzyme is important. We discussed about the antidotes, their mechanism, different anionic and serine binding sites we have discussed. Okay, I have listed out the classes. I have discussed about one or two about warfare agents also, their mechanisms. There also they will be asking questions. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. All the best for your exam.